Hi, my name is Casey, and this is Crash Course Biology, Enzymes Edition. So what are enzymes? You've probably heard of them at some point or another, whether on the labels of cleaning or beauty products, or in respect to dietary supplements. Enzymes are actually a type of protein, and like all proteins are made up of a sequence of amino acids. They also have a tertiary or 3D structure. Enzymes have two main functions, working as a catalyst for reactions and an agent for breaking down molecules. Enzymes catalyze or speed up reactions by reducing the amount of activation energy needed for the reaction to occur. In a catabolic reaction, where the reactants are coming apart in some way, the enzyme weakens the hydrogen bonds, which makes it so that the reaction doesn't have to spend as much energy breaking those bonds later. In an anabolic reaction, the opposite is done. The enzyme begins to take the reactants and makes it so that the hydrogen bonds begin to reform, and thus creating the resulting product. Without the enzymes, these reactions would take a lot longer. Some reactions that would normally take days are reduced to a few hours. Enzymes just make everything more efficient. First off, only one certain enzyme can interact with one certain substrate. To clarify, a substrate is something that interacts and is affected by an enzyme. The way it works is the substrate binds to the active site of the enzyme and is then affected by the enzyme as is needed, whether that be by loosening the bonds or strengthening them. After the enzyme has done its work, the product is released into the wild. All enzymes can and most likely will be reused. It's not a one and done deal. Enzymes continue to work as long as there is a substrate to catalyze. Enzymes also don't add any energy to the reaction. They only make it so that less energy is required for the reactants to react. The first type of inhibition is competitive inhibition. This happens when a compound which looks exactly like the substrate binds to the active site of the enzyme and prevents the actual substrate from being attached. This prevents the enzyme from performing its function, which results in less product being created. Also important is the fact that the competitive inhibitor is not broken down, so its only function is to keep the enzyme dormant. This isn't permanent, however, because the inhibitor can be overcome by an increase in the substrate. The second type of inhibition is non-competitive inhibition. Creative, right? A non-competitive inhibitor binds to the allosteric site of the enzyme, not the active site. It changes the shape of the enzyme so that the substrate can't bind to it. This therefore prevents more product from being created. A non-competitive inhibitor is most times the product of a reaction that is repurposed as an inhibitor. This happens when the product is in excess and needs to stop being created. This type of inhibition is called negative feedback. Cofactors and coenzymes are used to assist the enzyme in binding to the substrate. It works by binding to the active site so that the substrate can fit. Cofactors are inorganic molecules, while coenzymes are vitamins. Enzymes in the body, for the most part, work to digest food and essential nutrients. Because of this, enzymes are an essential part of the body's metabolism. Gastric enzymes are used in your stomach to break down food. DNA helicase is used to separate the two strands of DNA so that DNA replication can occur. And restrictive enzymes are used when cutting parts of DNA in bacteria. People cannot live without enzymes because the reactions would take too long on their own, and the body wouldn't be able to keep up with the amount of nutrients that's being taken in. Enzymes are proteins, so their hydrogen bonds can be denatured by a few factors. One is pH. An environment that is too acidic or too basic can denature an enzyme. Really high temperatures also can break the hydrogen bonds within the enzyme, causing it to denature. Enzymes can, however, adapt to their environment to continue performing their functions. As previously mentioned, gastric enzymes have adapted to the acidity of stomach acid so that they can function properly and break down food. Today you learned what enzymes are and what they do, the specifics of how they work, their application to the body, and how they denature. This has been Crash Course Biology, DFTBA.